Hello everyone. Okay, so upon your request, I'm here to conduct this class for your molecular basis of inheritance. Okay, so our main discussion today is going to be the biochemistry behind genetics. See, rather I tell this to all of my students, in most of my classes, that biochemistry is something inevitable when you're talking about in biology. Rather, chemistry itself is a part of biology. Just the way we say mathematics is the language of physics. We often say this, right? Similarly, I can say there is a very it's an unbreakable relationship between chemistry and biology. So, allow me a minute. Okay. Great. So, as I was saying, when you're talking about biology and chemistry, it is as if you're studying biology and you call this the study of life, then I would say chemistry is life. Right. So basically, in biology, you're actually studying chemistry behind everything. You can ask me anything, or you can give me any biological phenomena, and I'll give you a chemical explanation of that. Because, again, I'm telling you, life itself began as a chemical. We all know this in evolution chapter, right? The inorganic origin of life. So we know that these are the chemicals which aggregated to form biomolecules, which actually started life. Henceforth, I can say that whatever you're talking about in life, whatever you're talking about in biology, I can trace a origin of that, an explanation of that in chemistry. You talk about respiration, you know very well what cellular respiration is, and it's a huge biochemical pathway. You talk about, say, locomotion, then again there is all these bond breakage that actually causes or releases energy for your locomotion. You talk about anything. You talk about the formation of the cell or the cell cycle itself. That's what we are going to study over here in molecular basis of inheritance. You can see that there is a huge amount of biochemistry involved in the breaking of nucleotides, right? Breaking of nucleotides and breaking of these genetic materials, their recombination, their replication, their formation, everything is dealt with biochemistry. Because I can say, in other words, life itself is chemistry. We can talk about life as a very basic fundamental unit, and that would be genetic matter. You all know this, right? That's how we actually define life. That's the reason why even there is a you know uh, confusion as to whether virus is a living organism or not, because it has got genetic material which will perfectly tell me that it is a living organism. Doesn't matter whether it is performing its life processes, doesn't matter whether it's actually showing any symptoms of living. Just because it has got these what do you say, genetic material, I can pretty much say that it is almost a living organism. Okay? So now when we're talking about these essential part of living organism, that is a genetic material, and again we know the genetic material itself is a bunch of biomolecules. It's a bunch of what we call nucleic acid. We studied this in last year, right? So let us just start our journey with a very basic thing as to what exactly is these genetic material. We all know about this, right? That when you're talking about, say, genetic material, right, it can be either DNA or an RNA. Now, what is the basic difference between the two? RNA is the fundamental structure, and DNA is the same RNA where I kick off the oxygen molecule. I take an oxygen molecule, I remove it. So I'm deoxidizing it, right? And when I deoxidize it, or when I reduce this, so DNA is a reduced form of RNA. RNA undergoes reduction. I'll tell you how as well. Good. Right. So So when you're talking about these, now in this chapter, basically our flow is something like that. We will study about DNA first, and then we'll study about RNA. That's it. That's the end of the chapter. There's nothing much over here. We have two different segments. Once we're studying about DNA, then we're studying about RNA. So let us straight away begin with DNA. We got nothing else to compare over here. Now when you're talking about DNA, so now the first thing you need to know is what is a DNA. I told you it's a reduced form of RNA, or it is rather a nucleic acid. Na stands for nucleic acid, right? Deoxyribonucleic acid. Now, when you're analyzing the name itself, you will get to know what it is because you will get to know the constituents of it. When I say it is deoxyribonucleic acid, right? Then I can tell you something. I can get some hints over here. Now, if you're very keen in biochemistry, if you really like to study biochemistry as a part of biology, then you will know that there is something called ribo over here. And I know something. I mean, I can flash out something when ribo comes into my mind, right? Ribo is a part. It's a type of sugar, right? It is a type of sugar. Now, deoxy tells me that this particular sugar is reduced. Okay. And nucleic acid. Now, what do I understand by nucleic acid? Now, again, nucleic acid has got its own components. See, nucleic acid is a biomolecule. Biomolecule, in a sense, is a very complex biomolecule. It's almost, you could say, a polymer. Not actually a polymer, but it will be very soon polymerized, and that is what DNA is actually a polymer of nucleic acids. So, what is this nucleic acid? Now, it's something like you can understand it this way. When you're constructing a house, probably you will need some raw materials. You will be needing cement, bricks, right, wood, iron, stuff like that. Similarly, DNA is a structure, right? It's a structure. You all have studied this, that spiral, helical sort of thing. So if this is a structure, it has got its raw materials. I cannot say it just comes into existence like that. That's very vague. So what we do is we look at its components. Now the components of DNA is very peculiar, and that is actually understandable by the name itself. Right? It has got the major portion of DNA itself is a sugar. Now, this sugar has got its own characteristics. It's not a simple molecule of sugar. Now, what is a sugar? Sugar is a bond of, or rather, I can say a molecule consisting of carbon, hydrogen, and oxygen. Right? C6H12O6, the most common type of sugar you know. Right? What is that? Glucose. Right? Do you know about fructose? Fructose is C6H11O22 or something like that. There are so many types of sugars. Now, we have already studied about glucose, fructose, maltose, like galactose. All these are like quite common to us. But these are the special names given to those particular sugars. Sugars have also got their own IUPAC names. Right? Based on the structure, we give them IUPAC name. And sugar is a biomolecule, so obviously it has got a name in chemistry. So depending on the number of carbon atoms, we either call it monosugar, a disugar, a triose sugar, a quattro sugar, a pentose sugar, right? Hexose sugar, right? Septose sugar. Right? We got so many types of sugar in that, right? Depending on the number of carbon atoms. So if you're talking about the sugar which is actually present in DNA, right? It is a pentose sugar. In a sense, it has got 5 carbon atoms. Okay. Now, if you got 5 carbon atoms, we can somehow, make a rough guess as to what the structure of this thing would be. Right? It's going to be something like this. Okay. Right. 1, 2, 3, 4, 5. Now, let me tell you, there is one more over here, one more strand over here. I'll tell you why, quite, very short period of time. Let us just forget that now. Let us just take the basic structure itself. This is what? This is a pentose sugar. Okay, and this pentose sugar, right? We have a specific name for this. Just the way we, say we have n number of hexose sugar. Right? Glucose is a type of hexose sugar. Right? Fructose is a type of hexose sugar. So similarly, this pentose sugar has also got a name, and this is ribose. Okay, this is or ribose. Ribose. You could call it ribose. Okay. So ribose is a pentose sugar. That is a very you can say major component of the entire DNA that we have. Now, apart from the sugar, you got other substances. The first thing is you got a phosphoric acid. That is H3PO4 2 minus. Now, this will tell you something about why we actually call DNA an acid. Like we call it nucleic acid. Why do you call it nucleic acid? Because of this phosphoric acid. Rather, I wouldn't say there is phosphoric acid present. There is actually a phosphate group present. Right? There is a H3PO4 group present. Right? Now, what happens is this group will then get a 2 minus charge, and I'll tell you how as well. That is due to the bonding in the DNA molecule, as we know it, and that will give us a acidic form of this phosphate group. It is just a phosphate group, but because of the negative charge, it becomes acidic. It starts releasing H plus ions, and that is how we call it. I mean, that is the reason why we actually call this nucleic acid, because of the presence of this phosphoric acid in it. Okay. So the next thing we have is a nitrogen base. Now, this is one of the most important components, I would say. So what is a nitrogen base? See, nitrogen base is simply defined as a molecule with nitrogen in it. That's it. Nothing else, nothing much complicated to it. It's just a biomolecule with nitrogen in it. Now, this biomolecule can be of two types, depending again on their chemical structures, right? So, usually nitrogen bases are hexagonal in shape. Okay, so this hexagonal shaped nitrogen base could be either in this way, right, or it could be in this way. You can draw a benzene ring like thing that probably that's going to be much simpler. What is the basic difference you see in these two structures? It is a single ring. This is a double ring structure, right? And these are the two types of nitrogen bases I have. Obviously, it has got a nitrogen at the bottom. That is the reason why we call this nitrogen base. Right. Now, due to this structure, right, due to this two distinct structures, both of them coexist in a single DNA molecule. We call the single ring ones as purine and the double ring one as pyramidine. Okay, we either call it as a purine or a pyramidine. Now, again, when you're talking about this particular molecule, purine, this could further be of different types. Depending on the location of nitrogen base, right, depending on the location of this functional group, depending on the location of oxygen, depending on the location of, I mean, I would say there is rather another uh, extra methane group in both the cases. So again, depending on the position of the methane group, whether it is ortho, para, or meta directing, whether the nitrogen is ortho, para, or meta position, it could be anything, right? Because we are talking about a very liberal molecule over here. Nothing is restricted. So it is further of different types. Okay, and that is what we'll be discussing now. I'll be having this part over here. So we're talking about purine. That is a single link structure. It could be of two types. Okay, the ones given to you is adenine and guanine. We often represent it by just capital letter A, but for your convenience, I'll be writing the whole thing. We have adenine and guanine. Now understand, both of them are purines. Both of them are purines. Both of them have a single ring. But depending on the position of these functional groups, right, whether we have a nitrile group or we have a oxo group, wherever it is lying in the ortho, para, or meta position, or maybe some other ionic forms, we have it into two types, right? Now you don't really have to know what the structure is, as in what is the structure of this adenine or guanine. You don't really have to know that. It's not of that much important. Okay. So that is the two types of purines. Now we represent this by A and we represent this by G. Two types of purines we have. Pyrimidines, we have another This is again of two types. Pyramidal is again of two types. You have cytosine and uracil. C and U. But there is a problem. When you're talking about uracil, this particular pyramidal, right? The rest is okay, fine, because they have just a number of oxygen molecules that we need. But we are calling this particular nucleic acid as deoxyribonucleic acid. That means one oxygen molecule has to be deficient. That is the reason why we are calling this deoxy. So this deficient oxygen molecule, now you are not in a status as to ask me why it is that oxygen is being diffused from uracil only and not the other nucleotides. I don't think you will even understand it if I explain you that. But as of now, just understand, oxygen is always kicked out from uracil. It is again due to the bonding. Now, again, I'm telling you, it's not a point to question what sort of bonding is that and why is it that oxygen is being kicked out only out of uracil and not out of the other ones. But we call this deoxy because one oxygen has to be removed, and this removed oxygen is always the one from the uracil. So what happens is, from uracil, I'm going to remove an oxygen. So this is basically my structure. There is going to be some oxygen atom over here. I cut this oxygen atom, I remove this, right? I deoxidize it. If I deoxidize it, then this uracil is called thymine. Again, note it. It is called uracil. It is called uracil. Thymine is a modified form of uracil. Thymine is a modified form of uracil with one deficient oxygen. Uracil, remove one oxygen, you get thymine. So I can clearly say that DNA has got adenine, guanine, cytosine, and thymine. What about RNA? RNA has got enough number of oxygen molecules. There is nothing deficient over there. So if you're talking about RNA, then I can clearly say that the nitrogen bases would be adenine, guanine, cytosine, and uracil, not thymine. Thymine is when one oxygen is deficient, that is in case of DNA. So in case of RNA, it is uracil. In case of DNA, it is thymine. Okay, clear about this? Great. So now these are all, this is what we call nucleosides for short. Okay, we call these nucleosides rather. Great, that is what we call it. And then you also know what are nucleotides. Nucleotides are the bondage of these. Okay, we already know that adenine bonds with cytosine and guanine bonds with thymine. Okay, now these two bonded forms, right? The what do you say? The combined state of these two. Right, that is what we call a nucleotide. As of now, nucleotide is not required. I'll get to that when it is required for you. But as of now, just understand these are the nucleosides and these are the basic classification of nucleosides. Now that you understood, now these are all the components or these are all the ingredients that we require to make a DNA. Now we have got all the ingredients. We have collected the nitrogen base, whatever it is, all the four of them, right? Enough, sufficient in number. We have collected the phosphate groups. Rather, we have collected phosphoric acid and attached it to all the pentose sugars, making it have a phosphate group. Right. And that's all we require, I guess. Nothing else. Yeah, nitrogen base, phosphate, and sugar. So we got all these. Like we're gonna mix it up, and we're gonna prepare a dish, and that is the DNA. So when you're talking about DNA, it is actually a bond of all of these. So if you recall all of them, then I will be able to show you the structure of DNA. Now this structure is quite debatable for your understanding as of now. As we proceed with the chapter, you will understand quite good what the structure is. As of now, you will be like, why it is like this? Why isn't it like that? Because it happens that now, as of now, you don't really know, the microscopic and the macroscopic structure. So you might be in a verse to question. But let me just try to make a structure for you. Okay. The first thing you have is a sugar. Sugar, I told you, this is an important component. So we draw it first. Okay. After this, you have a nitrogen base attached to it. Okay, so let us consider nitrogen base as adenine. It could be anything. It could also be guanine. It could also be cytosine, thymine. Doesn't matter. We consider adenine usually. This is considered as a prerequisite. You could say. Okay. Then we have a phosphate group. A phosphate group H3PO4 2 minus. Okay. Clear up to here? Right. Then we have another methane group, I told you, another methyl group that is being attached. You will understand why is this a methyl group. You will understand it in quite a few seconds now. Now, always remember this. Now, this is a very important thing for again for you to understand. Always, adenine binds with thymine, and cytosine binds with guanine, or guanine binds with cytosine. I guess. Always it happens in this way. Again, why? Because they are compatible. That's what I can tell you right now. I can't really explain the mechanism of the formation of these bonds. I can only tell you that adenine cannot bind with guanine and thymine cannot bind with cytosine. It's just not possible. They are not compatible. Like hydrogen cannot bond with neon and form neon hydride or something. It just doesn't happen in chemistry. Okay. Now, if these two are binding, there might be some binding forces between them. Like it could be a van der Waals force, it could be a polar force, a London force, or a dispersal force. So over here, the force is a hydrogen bond. Very weak, right? You might be thinking the same way. But let me tell you, these hydrogen bonds are not, the simple hydrogen bonds we see in our regular inorganic molecules. In case of inorganic molecules like hydrogen fluoride or maybe water molecules, we see hydrogen bonds are quite weak. There's just a single bond between hydrogen and oxygen. Over here, we're talking about a highly electronegative component, as highly electronegative as phosphorus, and we are binding it with hydrogens and carbons. Just imagine, this is a very, very strong hydrogen bond. Not even a hydrogen bond, I would say hydrogen bonds. Now, that is a very important statement I gave you right now. Because of this multiple bonds, we have DNA actually existing. That is the reason why DNA doesn't get denaturated at your blood temperature. Otherwise, DNA would have boiled and broken down. The bonds would have broken down. It is still that hard because of the type of bond that we have, and these bonds are quite unique. Adenine and thymine have got two hydrogen bonds, and guanine and cytosine have got three hydrogen bonds. Just imagine three bonds that would hydrogen bonds. It is quite a strong force. Trust me. Although it is hydrogen bonds, having two or three hydrogen bonds are as good as having something as maybe strong as polar induced polar bonds. Okay, so this is a very strong force of attraction that we have. So I can tell you something, that breaking of these are impossible, almost impossible. There's only one thing that can break these bonds. Okay, and you will study it in the very next part of this chapter, and that is ligase enzyme. There is an enzyme called ligase that actually breaks these bonds, and because of them breaking the bond itself, DNA is being replicated. You will study that, don't worry. So obviously, this is bonded with thymine. Now, thymine, this is another nucleoside out of a part itself, right? I mean, thymine is not a part of this. So this thymine has got its own nucleotide. And what does its own nucleotide consist? Again, a pentose sugar. Now the rest of it is all common for everybody. Okay. H3PO4 2 bonds. Right? That's the structure I have. Now again, we've got two more, nitrogen bases, right? Let us utilize these as well. Now again, we have another sugar molecule. Right. Again, it has got a nitrogen base H3PO4 2 minus. Again, it has got a guanine this time, triple bonded with cytosine as I told you. Then again a sugar. Now think about this. Phosphorus we have here. We have oxygen, electronegative, highly electronegative. We have hydrogen over here, which is electropositive. Again, phosphorus, hydrogen. Don't you think there's going to be some bonds? Of course there will be, right? And that is why we have a bond like this, right? And that is the reason why we are actually forming a chain. Otherwise, DNA would have been pieces of these nitrogen bases. It would have been fragments. They are chain because of the formation of this phosphodiester bond. This is called a phosphodiester bond or a phosphate bond. Very important to understand. Okay, this is a hydrogen bond. Questions are often asked on this. Phosphodiester bond. Phospho, there is a phosphate group. Di, there are two ester groups. Ester or CH3, or whatever the ester's formula is. I don't remember it now. Right, so there is two ester groups binding with a phosphate group, and that is why we call this a phosphodiester bond. Now, you might see that the back of this pentose sugar is binding with the back of this pentose sugar. Similarly, back of this is binding with the back of this. That's why we call this a back bond. Now don't ask me why is that a dumb name, but it is just called that way. It is called a backbone. We either call this phosphodiester bond or we call this backbone. Now tell me which bond is responsible for the helical structure of your DNA. Is that the hydrogen bond or is it the phosphodiester bond? Okay, now if you don't know what a helical structure is, I will define that to you as well in just the next 1 minute. Don't worry, and then you will be able to answer it. Rather, you should be able to answer. Now again, this is a polymer. This is a polymer. So if it is a polymer, then we have chains of this being developed. So again, this thing will extend over here. We have another phosphate group bonded with another pentose sugar, adenine, thymine, like H3, PO4, 2 minus. This happens. This is a huge chain, like an infinitely long chain. Okay? Right. Now, whatever structure you've seen over here, right? This is just a microscopic structure. This is the structure of the bonds. If you want to see this in a macroscopic world, not a macroscopic world, still highly microscopic, but still a microscopic world with a defined shape, where you are ignoring the bonds and looking at the overall bond structure, whatever you call it, it could be the uh, Vesper structure or whatever. Okay, so this structure of DNA is found to be helical. Now, what do you understand by the term helical? Helical means spiral. You can simply call it spiral. It's not anything different. Just another term for spiral. So you have all seen this, right? This is something which you're all familiar with. DNA, right? Now again, I can tell you that this has got two strands. Right? You can call this as the first strand, and this is the second strand. Right? Two strands are present. The first strand is nothing but this region over here. Right? This region, okay, that is got adenine, guanine. That is being repeated in that way. It's got a sequence. And this strand, like the second strand, is got thymine, cytosine, and that sequence continues. So these are two different strands that you have. This is a strand, this is another strand. And you bond these two, automatically you can see that some of the bonds over here, some of the hydrogen bonds formed, right, cause spiraling of these molecules to occur, and this spiraling will give you some nodes, as we call this. Right. This is nothing but a point where the hydrogen bonds happen, and due to this strong hydrogen bonds, we have spiraling of the entire nucleic acid taking place. Okay. So again, the regions over here is nothing but our adenine and thymine and stuff like that. So this would be maybe adenine, right, and this would be maybe guanine, right. That's what it is all about. Every bar that I'm drawing over here is nothing but bonded nitrogen base. Here, that is the structure of our DNA. Now again, there are a little bit of specifications that you should know. We have a central axis about which the spiraling actually happens. So we have a central axis passing through all of the nodes. Right now, this is not that straight, but just imagine this to be a really straight axis, right? So with effect to this axis, you would say we have the diameter defined for the entire DNA molecule, and the diameter would be 20 angstrom, A naught or angstrom. So I can say 1 angstrom is equal to 10 raised to the power minus 10 meters. Just take it that way if you even require. Okay. Then between these two nitrogen bases, right? Between any of the two nitrogen bases. So this distance is calculated to be 3.4 angstrom. Okay. And from the central axis, right, from over here maybe, this distance is calculated to be 10 angstrom. Obviously, I have 20 angstrom as a diameter, and this looks like a radius to me, so obviously it is 10 angstrom. Now this 10 angstrom is called a shallow groove. Okay, we call this not a shallow groove, we call this a deep groove, because these are the shallow grooves that we have. Okay, so let me just name it first. This is a deep groove, this is a shallow groove. Now, on what basis are these deep and shallow? This has got a huge, or I could say this is equivalent to the radius of the entire molecule, so we call this deep groove. This is much less than the radius, so we call this a shallow groove. Okay, so the diameter of the shallow groove is much less than that. Diameter of the shallow groove is given to you as, so it's not even given to you, it's much negligible, you could say, right? And then the radius of the deep groove is 10 angstrom. Now another thing you should know is between two nodes, like I have two successive nodes, right? Now between them we have 34 angstrom. So this is one spiral. This entire thing, right? This is 34 angstrom. Think about it this way. So this is one single spiral. Like we have one H shape. This is one spiral, one strand of DNA, you could say, one spiral of DNA, or one helical of DNA, you could say, and that will be of 34 angstrom. Now, all these measurements are not required for you. Just I am giving this as an information for you. It's not at all asked in your exams, hopefully. Okay. So we have discussed so far about the basic structure of DNA. Now, there is a little bit more to add to this, and that is the Watson and Crick's model of DNA. Now, this is what the Watson and Crick's model of DNA is, right? So, what are the properties? It says it's a double helical structure made up of two strands. That it is well defined on our chemical, molecule basis, right? And then he has defined all these distances, right? What else has he defined here? That's it. That's all it has got to do with it. And the linking bonds. What are the linking bonds? Again, these bonds are, as we know, the hydrogen bonds, right? And these bonds are, as we know, the phosphodiester bond. Okay. Right. Now there's one more little theory again, that is Irwin-Chargaff rule. Now Irwin-Chargaff rule is not a part of your NCERT syllabus, but rather an important part in the NEET and KVPY syllabus. KVPY 2015 and 2013, I've seen two questions based on Irwin-Chargaff rule. Now Irwin-Chargaff rule states that A plus G upon C plus T is equal to 1, or rather, now what are these A plus G? Again, you know what are A, G, C and T, right? Now the concentration of concentration of adenine plus the concentration of guanine is equal to concentration of cytosine plus the concentration of thymine. This is what the Irwin-Chargaff rule states. How is it true? Because we know that adenine always binds with cytosine and guanine always binds with thymine, right? So these two are always and always bounded. Made a mistake. Adenine always binds with thymine and guanine always binds with cytosine. Since these are always associated with each other, they never just break up, right? And that is the reason their sum or their calculated concentrations are always equal. Again, this is used in solving questions. You'll be asked questions like maybe there is a say there's a DNA or genetic material which has got maybe 10% of adenine, then what is the percentage of cytosine? Then you should be able to tell that if there is 10% of adenine, then obviously there is another 10% of thymine, because there is these two are bonded. So there will be 20% of adenine and thymine. Now the remaining is guanine and cytosine, that will be like remaining 80%. And out of 80%, cytosine itself will be 40%. So that is where you're going to apply Irwin-Chargaff rule. If you didn't understand what I said, I just rewind the video and say it again. Rather, I'll tell you this is quite important. Okay, although I went a little fast. Then you have some, introduction to who found out DNA. Like the term was given by Zacharis. Okay, and then you have Harris, who has done the hybridization of DNA, right? And then DNA was first extracted by Friedrich, and the nucleic acid term was given by Artman in 1899. So these are like some theoretical questions which I cannot teach you. All you have to do is learn it. Okay. Coming to the next part of your DNA. Now there are another like a few terminologies that has been given to you in NCERT. Other following this book right now, right? The LLS, but in the NCERT they have given you these terms, that is promiscuous DNA, right? Repetitive DNA, satellite DNA. What is repetitive DNA? This particular sequence, right? It is highly repetitive. Now you will not be able to understand this unless I explain you what is a RNA or what is a codon. Because if I understand, if I explain you what is a codon, then probably you should be able to understand the term repetitive. But as of now, I'll tell you that these stuff, like the adenine, guanine, or whatever we saw right now, these are bonded in a sequence. Okay, it is always like this. Usually, A G C T C A A G G T C T T. Something like that. Some random code. Okay. Now this always exists in codes of three, right? They are triplets. So this is a codon. 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 Stuff like that. Okay, it's always in triplets. Now, you can say that functions or whatever happens in our body happens because of these codons. Okay, there are some scanning mechanism in our body which scans through these, and whenever it finds AAG, it'll just stop it over here and cuts the DNA apart. This is called a nonsense or a stopping codon. Many codons are existing like that. Okay, you will study that in like maybe next video of this chapter. So as of now, what you need to understand is when you're talking about these codons, they can be highly repetitive. Highly repetitive in the sense it could be like AG, 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 C, T, AG. It might go on like this. Now I can see that AGA, like, and GAG, AGA, GAG, it is a highly repetitive codon. Now these are found in advanced organisms. This is an advanced sequencing technique. Okay, and this is what we call a repetitive DNA, seen in humans and complex organisms. Satellite DNA, DNA which actually runs around here and there. What we call jumping DNA. Okay, it could be many. You got many terminologies like that. Then there are some characteristics of DNA that has been given to you. That is denaturation of DNA or melting of DNA. Now, what do you mean by denaturation? Denaturation, the word simply means the cutting of DNA or the excision of DNA. Or I could say it is the breakdown of DNA. So I'm breaking down a molecule. Obviously, I'm breaking down into simpler parts. I'm fragmenting the molecule. If I need to fragment the molecule, then obviously I need to, you know, break some bonds out here. So in denaturation, these bonds are broken up. Okay, and also these phosphodiester bonds are broken up. So you basically have, after denaturation, you end up getting this type of strand. That is, one strand becomes two strands, and that too they are completely separated and they are not even helices. Okay, this is what we call the denaturation. This happens with high temperature when we are applying high temperature. So 90 degrees Celsius is the denaturation temperature for human DNA, where hydrogen bonds break down. Okay, that is G triple bond C and 8 double bond T break down. That is what we call that denaturation. And again, there is renaturation of DNA also taking place. Now, renaturation is not a spontaneous process. It requires some enzymes, right? And these enzymes are again called DNA polymerase because they're polymerizing the DNA, molecules or whatever you call it. Okay? So renaturation also happens. It happens at room temperature. You don't need to do anything else. Just add a few enzymes, and the enzymes will do the work. They will, they will form again. Now, this is a complex thing, but they will form something called a Okazaki fragment, and these Okazaki fragments will come and join the parts wherever there is, you know, uh, breaking happening. Okay, and then the rest of it is again added up by primers. So you get one full DNA and another full DNA. This happens in room temperature. It is the renaturation process of DNA. Great. So we have completed the first part of your DNA. That is the first half of the chapter, DNA, and in that we've completed half of it. So it's like basically covering a quarter of the chapter. So now, after 2 more minutes, I will be taking care of your evidences of DNA, right? And after evidence of DNA, we'll look at the DNA replication part. Okay? Just allow me a minute. You can note down whatever is being written in the board. So one more thing now, when I did this in live class, it took me 4 classes, 1 hour each, to just complete this much. Just whatever I did now, it took me 4 classes to complete this. I'm quite happy now that I'm doing this in what, 40 minutes? That's quite fast. At least for me, it is like breaking the record itself. Do you know one more thing? That this chapter is the most important chapter in your biology. Right? So this is a chapter which has got maximum weightage. You can analyze your NEET papers, your KVPY papers, and there are like so many questions based on your there's molecular basis of genetics. A lot of questions I have seen, especially in KVPY, का मतलब पूरा ऐसे stream, पूरा जो 2 mark question है, it is all about the same chapter itself. And biochemistry is quite important, so you cannot ignore this. Right. So now we will start with the evidences of DNA. Now, you need to understand one thing, that none of us actually knew what is a DNA. When Mendel was performing his experiments, when he came up with his Mendelian theory, right, or maybe later on when Darwin came up with his Darwinism, or maybe much later on when Lamarck came up with his Lamarckism, right, all these Mendelism, Darwinism, Lamarckism, all these scientists or biologists, whoever observed inheritance in genetics, did not know what a DNA was. DNA is a much modern, you can say, part of your biology. It was discovered much later to all of these experiments. So in that case, I can tell one more thing, or I can conclude another thing. Mendel had no idea DNA is, or whatever that genetic material is. So how did he actually, call upon these? He called these Mendelian factors. Okay. And this has been going on for ages. Many scientists have been calling these Mendelian factors. It was known as Mendelian factor for a huge amount of time. I have already told you that the term DNA was only given in the year 18 or maybe 1920s. I don't know, it's not given in my book as of now, but somewhere in the middle of 1920s. So until then, this term DNA itself was unknown, and there was no information given whatsoever that genetic material is actually a chemical, or rather a biomolecule in structure. Okay, it was some sort of magic for everybody, or somebody thought there is no existence of genetic material. Genetics happens; it's a process, and there is no physical existence. And some people are, there is a physical existence, but that is actually the organism itself. The whole organism itself is a genetic material, right? And some people thought it was all pure magic, right? All the spiritual beliefs that you call it. So those who believe that there is something called, something that exists which causes all of this inheritance and evolution to happen, those people always prefer to call it Mendelian factor, right? Now, much later on, you see that some scientists are there who have made out experiments and proven that there is an existing chemical that actually causes all of this inheritance to happen. Now, this chemical was further, like much later on, was studied and discovered to be DNA. So, if you're talking about the very history of this particular biomolecule as we know it, this comes out completely experimentally with no basis, right? It was completely hypothetical until few of the good scientists have made experiments and came up with the hypothesis that there is a chemical compound that actually causes inheritance to occur. And out of this, we have two major biologists. That is, we have Liederberg and Tatum. Okay, Liederberg and Tatum who experimented with the mutant strains, and we have Griffith, one of the most important biologists, or one of, I could say, the second father of genetics after Mendelian is Griffith. Okay, whatever comes in genetics has to be got to do with Griffith, a major contributor of this field. And Griffith has made a very significant experiment which is seen nowadays everywhere. Whenever you talk about biology, what is the first thing that comes into your mind? Be frank, is it, don't some of you think that it's just mouses? Right? White mice, you cut the tail grows again. Biology. Right? But this is what we are actually picturizing biology based on, because Griffith's experiment was a political success. When Griffith made up this experiment, it was an attractive experiment, first thing. Second thing, even those people who didn't use science could understand this because it was a phenomenon which was well associated with people. Okay, and another thing, or the third thing he did was he was obviously I told you, right, he was a political person. So when he made up his experiment, it was like it was a tremendous success in the field of genetics. And his experiment was quite simple. He just poisoned rats. Okay, he just poisoned rats. But this was not as simple as that. Now let us understand this in detail. He took three mouses. Let me call this mouse A, mouse B, mouse C. Right. Here are some mouse like that. Right. And mouse C. Right. I got three mice. Now making this a little in the middle. Now, out of these three mice, what he did was poison. He was fond of killing mice, you could say, but he poisoned all of these mice. Okay, he poisoned it in a very specific, it's a very biological way, right? A very scientific method. He took a virus. Okay, this was a retrovirus, and it was well known at that time. It was the thing which caused. It wasn't really a virus. It was Diplococcus pneumonialis. Now this was something that caused pneumonia. Okay, I'll write it over here. Seen this question in need for a number of times, so better remember it down. So this particular bacteria, which was coccus bacteria, when we mean coccus, we mean they are coma shaped, and then it is diploid, diplo, right, and pneumonialis. It causes a disease based on the name of the bacteria itself, and we call it pneumonia. The pneumonia kills every single organism. Now there are two different varieties of Diplococcus pneumonialis. The first variety was virulent, okay, and the other one was non-virulent. Now, the way I remembered this in like for my exams is in this way: virulent bacteria are violent, and non-virulent are non-violent. That is what they are actually. Virulent bacteria causes pneumonia, and non-virulent bacteria doesn't cause pneumonia. It has got something to do with the structure actually. Virulent has got a textural structure, and non-virulent has got a smooth shell. Okay, this is something which causes resistance to antibiotics. Antibiotics or antibodies, you could say. Okay, so whenever this bacteria enters our body, our immune system gives a response. Like it produces immunoglobulins. Same thing happens with mice. Mice are also what do you say? Mammals itself. Right. So when this enters the body of say a mice, the mice also produces antibodies. Maybe it can be IgG, right? IgM. All these antibodies are produced. Okay, and these antibodies and these also release antigen. Okay, so these antibodies and antigen combine, and it forms a complex. And this complex or antibody antigen complex is harmless. It does not cause any disease, and that is what our immune system basically does. But what about these smooth shells, right? These smooth shells are what we call virulent. Okay, I made a mistake in the beginning. I called this virulent, right? This is non-virulent, and this is virulent. Okay, smooth ones. So smooth ones, they seem quite smooth. Okay, but they aren't actually. These things, right? These are the ones that cause your pneumonia to occur. These smooth shells have got some resistance. Now, these resistance does not allow your antibodies to attack these shells. So since antibodies cannot attack, it cannot bind with antigen and form antibody antigen complex. So complex formation, right, it's not possible. Or what we call agglutination. So agglutination is not possible in case of smooth cell bacteria. This happens with almost every single bacteria that is there in us. Whatever is infectious, whatever causes disease is of this category. Okay, whatever doesn't cause disease of this category. This cannot do anything. All it can do is reproduce. Hardly take our nutritions maybe. But mostly this will be killed out. Okay, whenever there is a antibody, just fuses with this, forms a complex and throws it out of our body. But whenever there is a smooth or a violent bacteria, these antibodies have a very difficult time to get into these bacteria and form the complex. So these are the ones that is actually the deadly bacteria. It could actually cause death even in humans as of now. Okay. Now Griffith, he took two samples or rather three samples of Diplococcus pneumonialis. Okay. Now the first sample that was injected in. Okay, I'll tell you what are the samples first. Okay, the three samples that he considered were the virulent ones, obviously, first thing. Virulent, alive. Virulent, killed, and non-virulent. Okay, so these are the three samples he has taken. Now, virulent, killed, and non-virulent, both of them cannot cause diseases to us. It cannot really harm us. Now, this killed in the sense this is actually heat killed, if you say. Okay? Heat killed. Heat killed in the sense again, that is the process of forming your antibiotics. In antibiotics, we take a bacteria, we kill it with temperature, or rather we extract the genetic materials at times, and we introduce it into our body. The same thing we do it with vaccines. Now what it does, as soon as it enters the body, it triggers the immune system. The immune system releases antigens, antibodies. Antibodies combine with these antigens. That is the end of the story. But the advantage is it gains some memory cells, and these memory cells or the immune cells in our body protects us from further infection of this particular bacteria. That is a basic mechanism, which is not really relevant over here. When you're talking about violent killed bacteria and the non-violent bacteria, these are peculiarly harmless. But these alive and violent bacteria, these are the ones that actually cause death in the mice, or the ones that is actually the causative agent of pneumonia in case of this particular experiment. Okay. Now what Griffith did was he took these samples and he shuffled it among these mice in such a way that mice A, for instance, had a virulent sample. Okay, this was a alive and virulent. Maybe over here he added a heat killed virulent. And maybe over here he added a non-violent. Now the results are quite obvious. When it is alive and violent, A dies. When it is heat killed and non-violent, violent, B doesn't die, right? B survives. And when it is non-violent, obviously there is nothing to fight with, so C also survives. Now he was curious. He did another experiment. Now this time he made another experiment of weird combination, you could say, right? Like mixing up all the ingredients. So what he did was he took another mice, the unfortunate mice D. Then this mice D, he injected the non-violent strains. Okay, non-violent was added plus heat killed violent. Okay, a non-violent strain and a heat killed violent strain is added. Now what could be the effect? Now the result of this particular phenomena itself, based on that result, Griffith made his conclusion that whatever inheritance is happening amongst us is happening due to the genetic material as a part of the chemical compound or the chemical composition of this genetic material. So what was the result? Now, some of you might have guessed it. Now what happens is, in case of your heat-killed virulent cells, right, these are killed, it isn't alive, okay, but it has got genetic material. The genetic material cannot be, rather you can say even it may be denatured, but it has got some genetic material, okay, and these genetic material, they suggest harm. What does the genetic material tell the bacteria to do? Kill the mice, right? Infect the mice with pneumonia. That's what the genetic material suggests the bacteria to do. Then we have the other ones, that is your non-violent. Okay. Now this thing is also got a genetic material. But this genetic material tells the bacteria to not harm, okay, or become harmless. So this genetic material is giving out information to the bacteria not to cause any harm, and these genetic materials are giving out information to the bacteria to cause harm. Now, when you're killing these bacteria, you're not really killing the genetic material. Now, what happens next is a very advanced stage, and that is something on the basis of which, or on the only basis of which, your entire, what do you say, biotechnology is, developed. Like the field of biotechnology is developed on the basis of the statement that we are going to tell you next. Okay, and it is quite simple. These genetic materials combine with these genetic materials. Okay, now you will say, how does it even combine? So there is something called recombinant DNA. You will be studying it in biotechnology. Okay, recombinant DNA, in short, we call this as rDNA. Okay, now the basic idea you need to know over here is you have a DNA strand. Okay, so this will give me, like, if I denaturate it, okay, I will get one strand over here and one strand over here. Now what I do is I'll eject this strand out. I don't need this strand anymore. I'll take another DNA. I'll split this into two. I'll eject one more, and I'm left with, now suppose this is strand A and this is strand B. Then I'll be left with one strand of A and one strand of B, and this will combine in such a way as to form that this will have a strand of A and a strand of B. This is a strand AB. Are you getting it? That is the basic idea of recombinant DNA. In our DNA, we take a DNA strand, denaturate it. Now, how do we denaturate it? Actually, we have something like a scissor. Okay, so this scissor will cut the DNA into two. Now, we don't physically use a scissor. Okay, but this is again all got to do with enzymes. We use lots and lots of enzymes over here. Okay, first we'll take the denaturase enzyme, then we'll cut up the DNA strands. Then take the ligase enzyme. We'll take one strand from here, another from here, add both of them, ligate or stick it, and it forms a rDNA. Again, there are something called restriction enzyme or restriction endonucleases. Now, restriction endonucleases, it search for some location, a specific location where it has to cut. Suppose if this is a DNA, it has got, say, A, D, C, C, T, A, D, D, some codon like that. So what is D? My gosh, G, G, G. Suppose it has got a command that it has to ligate in this region. Say wherever there is 2C, it has to cut it. So over here there is first restriction. It is broken up into two. So we have AGC, we have CTAGG. Then again, this thing is attached with some other maybe codon, say TCAGG. So these two are added, and then we have a ligase taking place between them. And this forms a recombinant DNA, AGCC, AGG. Something like that happens. That is a very fair idea I can give you as of now, just for your understanding. Okay? Now that's what is happening over here. So we have a harmful strand, and one strand of this harmful, virulent bacteria is recombined, or recombinant DNA is formed with a harmless strand of your non-virulent bacteria. So what happens next is that the harmful characters get expressed. But this is not your Mendelian expression or anything, but it just happens that way, that when you are forming a recombinant DNA out of the DNA of the virulent bacteria and the non-virulent bacteria, and then inserting it into any bacteria, virulent or non-virulent, the resultant bacteria will behave as a virulent bacteria. And that is what happens. Although this is heat killed, this isn't alive. It has got genetic material which it transfers into your non-virulent bacteria. Okay, and here it is. Now this thing is actually a recombinant DNA. As soon as you mix both of them, it forms a recombinant DNA. Okay, and when this thing reproduces, you get two bacteria, and surprisingly, both of them are virulent and not non-virulent, because the mother cell has got an infection of virulent DNA nuclease or a virulent DNA strand. So that is being infected to the mother cell. So both the daughter cells will express virulent characters, unlike the mother who had expressed a non-virulent character. Okay. So that is what happens. Now this DNA strand over here in the daughter cells will be virulent, and when these reproduce, you will get more virulent. Then virulent propagates and non-virulent decreases. So ultimately, although you are injecting only the alive samples of non-virulent bacteria, even if there is a single genetic material that is, a part of your virulent bacteria, then ultimately the DNA is going to form a recombinant structure, and a recombinant structure will express characteristics of a virulent cell and not a non-virulent cell. And that is what happened to mouse D. So mouse D actually died, right, instead of surviving. The death of mouse D was because although there was non-virulent bacteria present, it had got a genetic material of the heat-killed virulent which formed a recombinant DNA. Now, what was the result of this experiment? That is something you need to analyze by yourself. I will not be able to give you a hint. All I can tell you is that physically the virulent was dead, but genetically the virulent was alive, or the genetic material of virulent was alive. And that is what gave Griffith a suggestion that it is not the entire organism that causes inheritance. There's only one thing inside of the organism that actually causes inheritance. And that one thing was later defined as a genetic material. It was extracted, right, and it was studied upon, right, and then we have a lot of further experiments. I'll be listing out 2 more experiments in the same class today, okay, and then we'll end it today, okay. Right. So the next, you could say, experiment, which also suggested a result, which was similar to what Griffith told us long back. So Griffith was like, master of all. He gave out a result which was like a prophecy to the geneticists. But then again, you need a lot of experiment to, make up a theory or a principle, right? You can't really take hypothesis and say, "Ah, this is a theorem." You cannot really tell that. To, to prove something, you need a lot of approaches. And that is what the further scientists did. So they were like basically contributors of Griffith's success. And one of them was Liederberg and Tatum. Achha. Now Liederberg and Tatum, right, they also performed an experiment. Again, they took bacterial strains. Now one was called the mutant strain A. The other was mutant strain B. Now, a little bit of backstory before you can understand this. Bacteria, right, they have a characteristic feature which, with respect to their nutrition, okay. Certain bacteria, okay, they have some selective nutrition habits. Or rather, you can say that if I take some bacteria and put it in, let us make this, more something that people understand of, okay. Let us take this with a macroscopic word. Suppose there is 2 kids, okay, you and your friend. Now what happens is, suppose I ask your friend to go and go to some hostel and live there. Now your friend is quite, accommodated to all this. He's quite, you could say, flexible to all of this. So when you ask him to stay in a hostel, he's going to, like, maybe 1 or 2 days he will say, yeah, this is very horrible. He's not adjusted to the hostel life. But maybe after a week or a month, he may get adjusted to that place. And later he will eat the hostel food. He will use the hostel, rooms. He will sleep over there. I mean, he's quite sleepy. He's able to attend all the classes. He's made a lot of friends. So he's quite adjusted to that place. So suppose you are not like that. Suppose you are a little unflexible, you could say. So if I ask you to live in the same hostel, with, when you're living in a totally different life in your house, maybe if I ask you to drastically change it and live in a hostel, maybe you will start complaining. You will say कि नहीं यार ये खाना नहीं खाऊंगा मैं. I cannot eat this. It is so saltless. यहाँ पे नींद नहीं आ रहा है. बिस्तर कितना मतलब सख्त है यार. I cannot sleep over here. The bathroom situation is horrible. I cannot do there or whatever. So you will not get adjusted over there. You'll say people, yeah, they are all strangers. How can you even speak to them? Let me just stick to myself. Why do I even need these people? सबसे अकेले रहो. Like you have no friends. Like you don't really get adjusted to that place. And ultimately what happens, you leave that hostel and go somewhere else. You say कि यार मेरे से ये नहीं होगा. Now bacteria also suggests that sort of you could say character when it comes to their nutrition, solely nutrition, because that is something extremely dependent on their energy. Okay. Now what happens, certain bacteria, like they have a very selective nutrient habit. Now the nutrition of bacteria is proteins. Okay, so let us speak in language of proteins itself. Some bacteria, they can exist in conditions of methionine, say thymine, say serine, say um, anything, right? Thrombin, right? Thymine, whatever the condition may be, the bacteria is able to survive over there. And maybe its favorite nutrients is not present, right? शायद कुछ यहाँ पे XYZ nutrient है which the bacteria love so much but it's not present. तो वो क्या करेगा? He'll take the nutrients from the surroundings and say it's okay. I have to survive over here, so I'll adjust. Take the nutrients, cook its own nutrient, convert the nutrient from the surrounding into the nutrient that it wants. So it will produce its own nutrient and eat it and survive. ये उसके साथ होगा. But there are certain bacteria ना जो हमेशा ऐसे कामचोर होते हैं. So it does not really adjust with the surroundings. So what happens is if the bacteria needs methionine, it has to be given methionine. अब उसके बदले कहो कि नहीं यार मेरे पास आज methionine नहीं है, you eat serine today. What will the bacteria say कि यार मेरे से नहीं होगा? The bacteria will die. It will not consume serine. Whereas in case of the A strain, you can see that the bacteria actually takes serine and say okay कुछ भी हो I'll adjust. He'll take serine and he'll cook it and transform it into methionine and eat it. But bacteria B will say कि ये मेरे से ये नहीं होगा. Serine दोगे तो खुद लग रहा है serine, I'll die. Now this, the phenomena that I told you right now. Okay, on the basis of this, Liederberg and Tatum took two samples, mutant strain A and mutant strain B. The mutant strain A was that flexible type, so it could survive in any nutrient environment. You give it any nutrient, it will take it, convert it into whatever it wants, take whatever it wants, leave whatever it doesn't want. That's fine with it. So it can survive everywhere, right? But mutant strain B is very specific. It needs nutrients X, Y, Z, right? All these it needs specifically. Right? If you're not providing Z, it won't survive. If you're providing instead of Z, you say yeah, Z के बदले I'll provide A. I'll not provide Z, I'll provide A. So it won't survive. कहता है कि यार मेरे से ये नहीं होगा. मैं मर जाऊँ better है, but मैं तुम्हारा खाना नहीं खाऊंगा. That is the sort of attitude that the mutant strain B is showing towards the environment. Now what happens is Liederberg and Tatum, they cultured both the you can say strains. So mutant strain B was cultured in its favorable environment. Mutant strain A was cultured in one of the random environments. Mutant strain A survived. Mutant strain B also survived because it was given a favorable environment. Later, they changed the environment of strain B. Now B was cultured in an unfavorable environment. Although this was unfavorable for A as well, A could have adjusted to the environment by producing some of the nutrients that it wanted. But B cannot produce its own nutrients. It is solely dependent and rigidly dependent on whatever is available and out of it whatever it wants. So what happened was when you're culturing B in an unfavorable condition, B obviously dies or the colony decreases. But the colony of A was again it was flourishing in that particular environment. Now again comes your heat kill situation. They took heat killed mutant strain A. Okay, so now this mutant strain A was killed somehow. यार खाना नहीं तो क्या? उसको मार दो, उसको जला दो, ठीक है? मर गया सभी strains. Now this strain was taken and that was mixed with mutant strain B in unfavorable condition. Now what will happen? Initially, the mutant strain B will die because it is in unfavorable condition. It cannot survive in unfavorable condition. But as soon as A was introduced into it, we saw that although A was killed, its genetic material was present. That formed recombinant DNA with mutant strain B. And now mutant strain B actually showed behavior of mutant strain A. That means later in the same B colony, if you are providing unfavorable condition as well, B will survive because B has got a recombinant DNA of A. This again proved that inheritance happens not because of the whole organism but because of some chemical compound present inside the living organism. Okay. Another experiment again shown by Hashi and Chase. Now they took phosphorus. Okay, now this is another very important development I can say in the field of genetics. Hashi and Chase, they had a little suspicion as to what was actually present in the genetic material. What exactly is genetic material? हमको समझ में आ गया कि genetic material is present that causes inheritance. But is it a chemical? Is it a structure? Is it a hypothetical compound, inexisting compound? Now Hashi and Chase wanted to check this out. So what they did, they took out. Now they what they did was they took strains or maybe certain colonies, right, and injected it with phosphorus 35. Okay, it was a phosphorus 35 environment, you could say. Okay, and another compound, another colony with sulfur 35. Okay, so phosphorus 35 and sulfur 35 were the environments provided to two different colonies. Okay, now what they observed was as the sulfur 35 consumed sulfur from the colony, right, it consumed this sulfur as a mode of nutrition or maybe as a mode of respiration, whatever it may be. Consumption of sulfur took place. And now when it was separated from the medium, we saw that no sulfur was present in the bacteria. How is it possible that bacteria is actually consuming sulfur, but still it is not present in the part of the bacteria? Rather, I can say that this was probably because bacteria was consuming sulfur as a part of nutrition and not merely as a part of something that it can keep with it forever. Okay? It is something like, for example, I give you a cup of milk and a cup of water. You will drink cup of milk and cup of water. After maybe 2 days, I'll check you. ये तो नहीं हो सकता है, but I'm hypothetically speaking, I'll examine your body and I'll see that there is cup of water which I gave you that is still present in your body. Maybe it is somewhere in your blood vessel, maybe it's somewhere in your liver or somewhere. But the cup of milk I gave you is already excreted. It is not present in your body because milk was given for a different purpose. You threw it away. Water was given for a purpose which was served in your body. So at least a partial amount of that water was still present in your body. The same happened with sulfur. Sulfur was not required. So it's like a cup of milk. So the bacteria consumed it as a nutrition, then excreted it as a nutrition. But in case of phosphorus, we saw that the bacteria which were cultured in this medium consumed phosphorus, and when you extract it from the medium and take it into a mediumless situation, the bacteria still had the presence of phosphorus. Now, if it still had the presence of phosphorus, then phosphorus was not merely a mode of nutrition. So there was something in phosphorus that the bacteria liked to possess forever. And this something that it could possess forever, much later on we got to know that it was DNA, because DNA had a phosphate group, right? It has a phosphoric acid group. So obviously DNA requires phosphorus to develop, so you will be requiring phosphorus in turn. So when I provide phosphorus to you, you're not going to excrete it, right? Or at least a trace amount of it, you will keep it with you forever, because you will be developing n number of DNA molecules from that phosphorus which I have provided to you. So if I scan your body, I will be able to find the traces of phosphorus which I gave you even 10 years back. But if I scan the traces of, say, something like calcium or sulfur, then that is not present in your body, because you have already taken and consumed whatever you consumed, you wanted to, and you excreted it out. Okay, that was the basic idea. So this gave Harshney and Chase an idea that the genetic material consisted of phosphorus. Then again we have lots and lots of experiments. Okay, now there is something called a semi-conservative mode of DNA replication. Now, DNA replication requires your knowledge of the actual mechanism of DNA replication, which I'll be discussing in the next class. So अभी के लिए बस ऐसा समझ लो ना, मतलब I can explain this to you. चलो अभी के लिए छोड़ देते हैं, ठीक है? I'll tell you this in the next class when I'm explaining you the DNA replication. So far we'll close the class today. Right, I have discussed about structure of DNA and discovery of DNA. The next thing we're going to do is mechanism of DNA replication, because DNA has to be replicated for the replication is basically the reproduction of the DNA molecules, and that is required for cellular reproduction or the cell division as we know it. So obviously when there is cell division, there is DNA replication, and we will be studying this DNA replication in the next class. Then we'll be studying the semi-conservative mode of DNA replication, and then conclude it with ribonucleic acid in the next class. Okay, we'll do all of this. Thank you.